Good evening, my dear students and friends. Myself, Dr. Ravikant Pathak, and I'm going to start your video lecture class. In the last video lecture, we have discussed about the different excretory organ and structure as well as location of the kidney. And today I will start new topic of this chapter, and the name of topic is the nephron, its structure, and different types of the nephron. So here you can see they are showing the nephron. So this is the chapter name, and here they are showing the nephron. So nephron generally composed of the two things. First is called the Malpighian body or renal capsule. So there is the Malpighian body or renal capsule generally found. This is the first thing, and another is the renal tubule. So nephron generally consists of two things, and in the last video lecture I have already told about that. So nephron, it is considered as a structural and functional unit of the kidney. And 1 lakh nephron, it is generally, 10 lakh, not 1 lakh, 1 million. 1 million means 10 lakh nephron generally found in the kidney, and this is innumerable. Okay. So these nephron have the two different parts. First is the Malpighian body or renal capsule. This is also known as the Malpighian body or renal capsules. And another one is the renal tubule. Okay. So Malpighian body or renal capsules, they have the two things. First is the glomerulus and another is the Bowman capsule. What is the glomerulus? You have to see there is the not like structure of the blood capillaries. This is called the glomerulus. Okay, and another is the cup shaped structure. This is the cup shaped structure which forms like this structure. This is known as the Bowman capsule. This is known as the Bowman capsule. So in case of the glomerulus, there is the two different veins. There is the two different artery. So one is the afferent and another is the efferent. So two different artery. One is enters, another is comes outside from the this glomerulus region. So which enters, that is called afferent, and which comes out, that is called the efferent. Okay. So this is the structure of the Malpighian body or renal capsules. Another is the renal tubule. Renal tubule, they have the four different parts: PCT, loop of Henle, DCT, and collecting ducts. If we'll check loop of Henle, this is the U-shaped structure, and these loop of Henle, they have the two things. One is called the descending and another is the ascending. If we'll compare between the descending and ascending, so ascending is the thick and descending is the thin. Okay, and this play very important role into the counter current mechanism. So we will see when the urine formation or urine filtration, so that time we will discuss about the detailed counter current mechanism. So this is the structure of loop of Henle, and DCT. This is the distal convoluted tubules and collecting tubules where the water means excretory products. So where the excretory products after release and from the collecting duct to ureter to urethra, urinary bladder and urinary bladder to urethra. In this way generally pass. So this is the structure of the nephron. Now we will see the detail about the nephron. So here we have already seen. So there is the two different types of the not types, two different structure. First is the Malpighian body or renal capsules, and another is the renal tubule. Now here they have given the detail about that which I was telling. So here you can see. First is the nephron. So nephron is the uniferous. This is also known as nephron, also considered as a uniferous tubules, and it consists of the two parts. So there is the two parts. First is the Malpighian body and another is the renal tubule. Okay, so one is the Malpighian body, another is the renal tubule. Now you can see first is the Malpighian body or renal capsules. This Malpighian body also known as the renal capsules, and second part is known as the renal tubule. Now the proximal end of which nephron forms a blind or closed. What means of the blind or closed? You have already studied in case of the vertebrate chapter. So invertebrate chapter. So that means simply you can say animal kingdom. So in animal kingdom, invertebrata, we have studied the digestive system. So there is the two different types of the digestive system. One is incomplete, another is the complete. So we have already studied. One is the incomplete digestive system, another is the complete digestive system. Incomplete, they have the blind sac. They have the blind sac. Blind sac means if suppose that this is the tube like structure, So this is the tube-like structure. Only one pore opened and another pore is the closed. One pore open, another pore is the closed. That is called the blind sac. Okay, so you will see. So one pore open here, one pore open, but another pore is the closed. So this is called the blind sac. So here saying the proximal end of each nephron, blind or closed, means one end open, another end is closed, and enlarged and double walled cup shape and the cup like structure which is known as the Bowman capsule. So now here you can say this figure is showing the cup shaped structure which is known as the Bowman capsule. Okay, and these cup shaped structure generally formed in the Malpighian body or renal capsules. So capsules, Bowman capsule in the cortex, and these Bowman capsules located in the cortex region, because you have to already studied the two different region, cortex and medulla. So cortex is the outer portion and medulla is the inner portion. So Malpighian body or renal capsule is generally located towards the cortex region, and they have given this capsule is known as the Bowman capsule based upon the scientist. They are English physiologist as well as histologist William Bowman. On the basis of William Bowman name, they have given such Bowman capsule name. Each capsule contains a network of blood capillaries. I had already told glomerulus. So blood capillaries, which is known as the glomerulus, which receives blood through the afferent arteriole, and the blood comes out through the efferent arteriole. And if you check the diameter, so diameter of the afferent arteriole is much. So diameter of the efferent arteriole is comparatively lesser. Afferent arteriole, you will see here in the figure. So this is the afferent arteriole, their diameter, and efferent arteriole. So afferent arteriole is the wide, their lumen is the wide, and here efferent arteriole, their lumen is the narrow. And due to the presence of narrow lumen, it will create the pressure gradient by which much maximum filtrate can be filtered inside the Bowman capsule. So Bowman capsule, it is saying about the Bowman capsule, and here you can see Bowman capsule and glomerulus receives, Bowman capsule and glomerulus receives about 20 to 25% of the cardiac output at the rest. Cardiac output means blood. So cardiac output means during the release of the blood from the heart, that is called cardiac output. And there is the formula, so which I have given, we already discussed. We will discuss later in the body fluids and circulation chapter. So you should to know Bowman capsule and glomerulus receives about the 20 to 25% of the cardiac output during the rest phenomenon. So this is the basic function of the Bowman capsule. Now we will see the second is the renal tubule. So second, here they have been. Before this renal tubule, I am going to discuss about the structure of nephron. So here you can see structure of nephron. Very easy. You have to already studied from the different standard. So first off, this is the again the afferent arteriole. This is the wide lumen, and this is the Bowman capsule. This is also known as the Bowman, and the blood capillary structure, which is known as the glomerulus. And Bowman capsule sometimes also known as the glomerulus capsule. Okay, and here this afferent arteriole, when comes outside from the glomerulus region, this is called the efferent arteriole. Okay, and here second set of capillary, and those regions of the tubules which is found near to the Bowman capsule, that is called the PCT, proximal convoluted tubule. And this is the U-shaped structure which is known as the loop of Henle. And this loop of Henle, I had already told, involves in the counter current mechanism. And one thing here also they have given about the ascending. So it will see, so this is the descending loop of Henle, and this is the ascending loop of Henle. Descending is the thin and ascending is the thick. So here why I had already discussed. After that, so some part of the tubules which is far away from the Bowman capsule, this is called the distal convoluted tubules. And after that, this distal convoluted tubules combines form a straight collecting tubules or straight collecting ducts. So here they have given the name Bowman capsule, glomerulus, Malpighian body, proximal convoluted tubules, Henle loop of ascending and descending leaf, distal convoluted tubules, collecting duct, afferent arteriole, efferent arteriole. So this is the different structure which is found in the nephron. Now we will see the renal tubule. Renal tubule, it is the second part of the nephron. What is the role of renal tubule? So here you can see the Bowman capsule opens into a proximal convoluted tubules at the anterior part of the PCT is more coiled. If you compare the anterior part of PCT, is the proximal convoluted tubule is much coiled as it's the posterior part. Means posterior part is the less coiled and anterior part is the most coiled. Okay, almost and posterior part less coiled and almost straight. The proximal convoluted tubules open into a Henle loop. It opens into the Henle loop U-shaped structure. The Henle loop is U-shaped, I had already discussed, and makes hairpin turn. It forms the hairpin turn like structure, which has a distinct descending limb and ascending limb. Okay, and descending limb, I had already told, descending limb is the thin and ascending is the thick segments. The ascending limb open into the distal convoluted tubules, and the DCT is a coiled structure. We have to see in PCT and DCT both are coiled. PCT anterior part is the highly coiled and posterior part is the less coiled and straight. Many distal convoluted tubules unit to form a collecting duct, and the collecting ducts of the one pyramid. This is the very important sentence you should to know. The collecting ducts of the one pyramid unit to form a duct of the Bellini. The duct of Bellini lead into the pelvis part, and pelvis opens into the ureter. Okay, and by which ureter opens urinary bladder and ultimately urethra. So that means here saying the collecting ducts of the one pyramid, because we have to see pyramid like structure. In between they have the collecting duct. So here the collecting ducts of the one pyramid. So this is the collecting ducts of the one pyramid. Okay, and they unite means unite to form a duct of Bellini. This is called the duct of Bellini. Okay, so this is the very important structure they have already asked in your NEET exam. Now, one is the Vasa recta. What is the role of Vasa recta? You have to see in loop of Henle and Vasa recta, both are cooperative in function during the counter current mechanism. Vasa recta and loop of Henle. So both are the cooperative in function, means both is the necessary for the counter current mechanism. So here Vasa recta, what is the Vasa recta? If you see, so with the help of figure you can easily understand. So this is the figure. When the efferent arteriole comes outside, this will become the form numerous branching, and this branching is known as the peritubular capillaries, and this peritubular capillaries covered to the loop of Henle. This is called the Vasa recta. So now here you can read the efferent arteriole of juxtaglomerulus nephron. This is called the GGN. Juxtaglomerulus nephron forms a peritubular capillary system around the Henle loop. It forms the peritubular capillary system around the Henle loop, which is forms a Vasa recta. So this is the pericapillary tube which is forms a which is around the Henle loop. This is called the Vasa recta. And each of the Vasa recta makes U-turn at the innermost part of the medulla, and most of the Vasa recta, because loop of Henle they much found inside the medulla. So Vasa recti also runs inside the medulla. So each of the Vasa recti makes U-turn and innermost part of the medulla and return to the venous circulation near the junction. After that they will combine and form the venous circulation. This is the venule. Okay, and venous circulation of the near junction of medulla and cortex, that is called the juxtaglomeruli. That is the juxtaglomerular region which is near to the cortex of the medulla as well as cortex and medulla junction. Clear? So this is generally found. This is about the story of Vasa recta. Now we will see about the next, and there they have given the efferent. What is the different? What is the efferent and efferent arteriole and peritubular? It is saying efferent arteriole, the efferent arteriole and peritubular capillaries technically constitute a renal portal system. They form the RPS, renal portal system. Means efferent arteriole. Efferent arteriole means those arteriole which arise from the Bowman capsule, and further the number of branching forms around the loop of inlay, which is known as the peritubular capillaries. And these peritubular capillaries technically termed as a RPS, renal portal system. In all amniotes as well as reptiles, birds and mammals have at this have a this not at this have a this renal portal system of efferent arteriole and peritubular capillaries. Means you can say amniotes. Amniotes means birds, mammals and reptiles. They form the such type of the renal portal system. The Malpighian body and most of the PCT, proximal convoluted tubules. The Malpighian body and proximal convoluted tubules constitutes the distal convoluted tubules and are situated in the cortex region, because we have to see in Malpighian body there is the maximum parts located in the cortex region and situated in the cortex. Loop of inlay and collecting ducts are found in the medulla region. So loop of inlay and collecting ducts which is found in the medulla region. Okay, so this is the detailed structure of the nephron. So nephron is the two parts. First is the Malpighian body or renal capsules and another is the renal tubule. Now we will see types of the nephron. So there is the two different types of the nephron found. First is called the cortical nephron, another is the juxtaglomerulus nephron. So it is the cortical and here juxtamedullary, sorry, not juxtaglomerulus, juxtamedullary nephron. Okay, juxtamedullary means it is located towards the near of cortical as well as medullary region, junction of the cortical and medullary region. So that's why it is called the juxtamedullary nephron. Cortical nephron is the maximum present. It is the maximum 80 to 85%, and here it is 15 to 25% present in the juxtamedullary nephron. So now we will see the detail about the first is the cortical nephron. So here they have given about the detail. So nephron are two types, cortical and juxtamedullary, with regard to their location in the kidney. So cortical that means cortical nephrons, which is maximum found. Their maximum portion located inside the cortex region, and juxtamedullary, their maximum portion located inside the medulla region. Cortical nephron forms about 25% of the total nephron, and they lie in the renal cortex and have very short loops, inlay, and that extends only little into the medulla. It means cortical nephron, this is maximum portion of the cortical nephron. It is located inside the cortex region, while juxtamedullary, so remaining there is the very less number, 15 to 20% are juxtamedullary nephron and have their Bowman capsule close to the juxta, the junction of cortex and the medulla. Why they have given the name juxta? Juxta means the junction, the place where the cortex and medulla meet, that is called the juxta. Okay, so juxta and these juxtamedullary neurons have very long loop of inlay. If you compare between the loop of inlay about the cortical and juxtamedullary nephron. So juxtamedullary nephron, their loop of inlay is the very large. So loop of inlay is the large and extending deep into the medulla. It will go inside the medulla. And this type of nephron is present only birds and mammals. This conserve the much water. This conserve the much water due to the presence of large loop of inlay. Okay, so this is the types of nephron. First is the cortical nephron and another is the juxtamedullary nephron. Now here you can see with the help of figure easily you can understand. So very simple explaining. This is maximum portion because this is the cortex region, this is the medulla region. In the cortex region, their maximum portion, Bowman capsule, loop of inlay, this is the very short, and there is the little part of the loop of inlay and their portion located inside the medulla region, and maximum portion located inside the cortical region. So this type of the nephron, you can say, such type of the nephron is generally called the cortical nephron. Another, this is the juxtamedullary nephron. Why we are saying juxtamedullary nephron? Because if we compare here, the loop of inlay is very large, and this type of the nephron, it enters deep inside the medullary region. So this is called the juxtamedullary nephron. And why we are saying juxta? Because this is the position where the medulla as well as, so where the cortex and medulla both will meet together. This is the junction, and these generally located near to the junction. They are Bowman capsule. They are located in the junction, so that's why it have given the name juxtamedullary nephron. Okay, so with the help of figure, I think everyone you can easily understand the difference between what is exact position of the juxta as well as cortical nephron, juxtamedullary as well as cortical nephron. Now, the cortical nephron control the plasma volume when water supply is normal. Means when cortical nephron generally active, when the water supply is normal, that time cortical nephron active. And you know very well, it is maximum quantity is present, 80 to 85% present. And here the juxtamedullary nephrons regulates the plasma volume when water is in short supply. That means in adverse condition. In adverse condition, that time juxtamedullary nephrons, they regulate the plasma volume ratio. They regulate the plasma volume. Okay, means it maintains the plasma volume when the water crisis, water short supply. Water short supply means adverse condition will come. That time juxtamedullary nephron will active. And when the water supply is the normal, so that time cortical nephron is the active. Okay, and you have to see, so maximum cortical nephron is generally formed. Now we will see the difference between cortical as well as juxtamedullary nephron. So very easy, because we have to already studied the types of the nephrons. So cortical as well as juxtamedullary, you have to see the difference. So that is the simply here glomeruli, and in the outer cortex region we have to see their glomerulus region maximally located in the cortex region. Here glomerulus are close to the inner margin of the cortex. It is found in the inner margin of the cortex, because this is the juxtamedullary nephron. This is the juxtamedullary, this is the cortex. So their glomerulus region close to the juxta means junction of the cortex and medulla. Short loops of LA, cortex short, here long loop of LA. LA loop extend only a short distance in the medulla, because this is found in the only cortical region. Here their LA loop are found in the deeper in the medulla. Vasa recti are absent, cortical medulla. So there is the vasa recti means peritubular capillary is absent. Here peritubular capillary is present. Okay, and they are most common 85% of the cortical nephron, and they are very less 15% of the cortical nephron generally formed. So this is the difference between cortical as well as juxtamedullary nephron. Okay, so here they have given. So thank you for watching, and everyone should share, subscribe my channel, and hit the bell icon for regular updates. So thank you again.